Welcome dear learners. I am Mrs. Priyanka Chakraborty, Assistant Professor of BCDA College of Pharmacy and Technology in the Department of Pharmacology. Today I am going to present a session on antimicrobial resistance. So first we should know what are the learning objectives. After completion of the sessions, learner will be able to know about the antimicrobial resistance, understand the factors responsible for antimicrobial resistance, know the types of antimicrobial resistance, understand the mechanism of acquired antimicrobial resistance, and last of all, apply the knowledge to prevent antimicrobial resistance. So first of all, we should know what is antimicrobial resistance. It is the property of microorganism. It mainly includes bacteria, viruses and other parasites. Microorganism stops the antimicrobial agents, blocking the effectiveness of drug from working. So in this picture, it is clearly depicted that the microorganism basically blocks the working capacity of the antibiotics or the drugs. There are several factors which are responsible for antimicrobial resistance. The first factor is environmental factor. It includes huge population, overcrowding, poor sanitization, spread of the antimicrobial resistance due to transport facilities. Second factor is drug related factors. First of all, over the counter availability of the antimicrobial agents. Second, increasing use of antimicrobial agent. And third, irrational fixed dose combination. The third factor which is responsible, that is patient related factor. It is first self medication, second one is lack of education, third one is poverty, and fourth one is poor adherence of dosage. Fourth factor which is responsible for antimicrobial resistance, that is prescriber related factors. Here it is overuse of antimicrobials, inadequate dosing, prolonged use of antimicrobial agents, and lack of current knowledge and training. Now we have to know what are the different types of antimicrobial resistance. Basically, there are two types of antimicrobial resistance. First one is intrinsic or natural resistance. It includes some microbes are always resistant to certain antimicrobial agent. This type of resistance does not pose specific clinical problems. Example of this type of resistance is that Mycobacterium tuberculosis, which is the causative organism of tuberculosis, it is always resistant to tetracycline. Acquired resistance is the development of resistance by the organism. It is due to use of antimicrobial agent over a period of time. This type of resistance pose specific clinical problems. Now, my dear learners, it is the reflection point. So, we have to think and pause the video, and we have to know what is the mechanism of acquired antimicrobial resistance. After you record it in your notebook, then unpause the video and go to the next slide. Basically, mechanism of acquired resistance includes a flux pump, target site modification, and enzymatic inactivation and destruction of the drug. Efflux mechanism basically plays a major role in acquired resistance. It basically occurs due to changes in cell wall proteins or translocation of plasmids. It causes a flux of the antibiotics to the outside of the cell. Example of this type of resistance is the resistance of Streptococcus species against macrolides antibiotics. The next mechanism is the target site modification. We all know that when the drug binds to the target, that it only gives the effect. So the antibiotic binds the target site in the microorganism and gives the effect, but in that case of antimicrobial resistance, here what happens, modification of target site reduces the binding affinity to the antibiotics. Basically, target site modifications occurs due to chromosomal mutation and enzymatic alteration. Modification of target site blocks the drug to bind with the target. Example of this type of antimicrobial resistance is resistance of S. pneumoniae to penicillin type of drugs. The last mechanism in the case of acquired antimicrobial resistance is enzymatic inactivation and destruction. First, we will go to the destruction of drug. What happens? The drugs bind to the enzyme and there is destruction, and another is drug bind to the enzyme and there is change in the drug, that is inactivation of the drug, so that the drug cannot bind to the target. Example of drug degradation is the beta-lactamase enzyme. When drug binds with the beta-lactamase enzyme, that is penicillin, when binds with the beta-lactamase enzyme, there is degradation of penicillin and hydrolysis of penicillin, and when there is acetylation of aminoglycosides by the N-acetyltransferase. When the aminoglycosides bind with the N-acetyltransferase, there is inactivation of the drug, that is the inactivation of aminoglycosides, and it cannot bind with the target. Now, my dear learners, we should apply our knowledge to prevent the antimicrobial resistance. As antimicrobial resistance is global microbial threat, we have to use antimicrobial in the judicious way. We have to prevent the use of antibiotics in animals. We have to avoid the antibiotic in commercial cleansing product, and last but not the least, if we can develop newer antimicrobial agents, it would be very good for the prevention of antimicrobial resistance. Summary. So, after completion of this session, the learners are able to get knowledge about the antimicrobial resistance. They can understood the factors responsible for antimicrobial resistance. They gain knowledge about the types of antimicrobial resistance. They can understand very well about the mechanism of acquired antimicrobial resistance, and also they can apply the knowledge to prevent the antimicrobial resistance. Thank you. And for any queries, you can contact me in my email ID.